B20 Chapter 10.1 today. 2 lessons in this chapter, so it's pretty quick. We're looking at structures of muscles in the human body for starters. And what we can take a look at first is a pair of antagonistic muscle pairs, and that would be something you can try right with doing a bicep curl and seeing that when your bicep is contracting, the antagonistic or opposite muscle pair, the tricep, is relaxing. So they work together, and this will be something that happens all over what muscles attached to your skeleton. So to move the bones of your skeleton, one has to contract while the other relaxes. When you straighten your arm, then it's your tricep that's contracting and your bicep that would be relaxing. So that's one kind of muscle group, the skeletal muscles, and how they work. And we will be examining all 3 types of muscles in the body. So first type, smooth muscles, are the ones that are make up your internal organs. So things like all your blood vessels, your digestive tract, your even tiny places like iris of the eye. These are muscles that sort of form sheets, and so they're long and tapered looking muscle cells, and it's interesting that they can work for long periods of time and not fatigue. The second type of muscles is the cardiac muscle, and that's specifically only found in the heart, making up the muscle of the heart. These cells are branched, and so if you think of the inside of the heart and how those sort of tubes of muscles appear on the inside, I always think of that when I look at the branched looking format of the cardiac muscle, because they're also branched looking. Just like smooth muscle, these are involuntary, which means we do not have control over the smooth muscles of internal organs, like for peristalsis, for example, nor do we have control over the cardiac muscle in the heart. Skeletal muscles, though, are the voluntary muscles, so they are ones that you will control in order to move your skeleton, and the muscle cells are given the word muscle fiber, and so that is the same as a muscle cell whenever you see that word. And there's about 600 different skeletal muscles in the body, so when you see these pictures of muscles, there's many to get to know. And these are also called striated muscles because of the striped looking appearance that they have under the microscope. So their cells are long, they're thin, and they have many nuclei in a muscle cell or fiber. Okay, those pictures would be similar to what they would appear like under the microscope. Smooth ones kind of look like a smooth sheet. The skeletal ones have that striping, and cardiac have that branched look to them. Skeletal muscles support the body and help the bones move, and do some protecting as well of organs and joints. And when one is contracting, the antagonistic one is relaxing. So you can say that the skeletal muscles are arranged in pairs. And throughout this PowerPoint, lots of interesting videos to take a look at, so do that. Structure of the muscle. The cell or fiber is quite long. It could be up to 20 centimeters long. So those cells are actually quite big and have multiple nuclei. They will be found in bundles of cells or fibers, and there's a little bit of connective tissue around that bundle. And we're going to look at how that structure looks in the picture that your textbook and notes have in just a second here. But there are blood vessels and nerves that run through the muscle and control the contraction and supply the oxygen and nutrients. This is a very important picture that you should know about, and we should start looking at the muscle as a whole. So this could be something like your bicep, and within that you have these bundles, like you see little bundles of cells and or fibers. Fiber and cell, remember the word muscle fiber equals muscle cell. And the muscle fiber itself is this over here. So this would be one cell or muscle fiber that is enlarged. And just like a cell has a cell membrane, there's a special name for cell membrane in muscles, and it's sarcolemma. Sarcolemma is the name of the cell membrane. And just like regular cells have endoplasmic reticulum or ER, those channels, muscle cells have the same channels, but they're, instead of calling ER, they're called SR, sarcoplasmic reticulum. And it's the same as regular cells ER. So those channels, you'll hear about them in terms of the area that stores calcium that's needed for muscle contraction. So we'll be hearing about ER or SR coming up. Now, what makes the components, the very important structural components of each muscle cell or muscle fiber, is these little myofibrils, which are sections of many actin and myosin myofilaments. So the filaments inside here, myofilaments, actin and myosin. The actin is these smaller beaded ones, and the myosin is the thicker red one here. The actin and myosin is what is the part that will sort of interact in order to contract and shorten the muscle and then open up and relax the muscle. So we'll be looking at how the actin and myosin function, but remember that they are myofilaments. They're found in a myofibril, and there are many myofibrils in a muscle cell or muscle fiber. The SR is the series of channels, and that sarcolemma is the membrane around the muscle cell or fiber. And your muscle itself is composed of many muscle cells. Okay, you guys should label that picture or redraw this picture so you know the components of muscle. So in these notes that follow, they basically explain a few other details about the structure, saying that every cell or fiber has around 300 mitochondria, which for a cell that is a large amount of mitochondria because of all of the energy needed to do the muscle contraction, that they have a lot. The chart here goes through those main structures that we mentioned. A cell is a fiber. The myoglobin is the same as hemoglobin, but it's the hemoglobin found in muscle cells. Still carries oxygen, just like the hemoglobin regularly does. Sarcolemma, we said, was the cell membrane. The cytoplasm, instead of calling it cytoplasm in a muscle cell, it's called sarcoplasm, just the fluid-carrying organelles. SR, psychoplasmic reticulum, is the same as ER, endoplasmic reticulum, those channels. Specifically, we will talk a lot about how it carries and holds and stores calcium for the muscle contraction process. Myofibrils are where the bundles of myofilaments are, which are made of the actin and the myosin filaments, which will interact, and we'll talk about those coming up. So these are all important structures of muscle. So when you look at a picture of the myofibrils, We will have to zoom in and talk about actin and myosin here so we know how the muscle contracts. And the actin is always these wrapped thinner structures, kind of like wrapped beads is what it looks like. And it's going to interact with this thicker myosin, and the myosin has myosin heads. So you can see these little myosin heads that are going to actually grab on and pull the actin as we examine the muscle contraction process coming up. Actin and myosin, here we go. There's how they are components of our smallest component of our muscle. And there is a chemical bond that will happen between the actin and the myosin head. The myosin will flex, as you can see here, and actually grab and pull the actin. So that's what will happen when a muscle contracts and shortens. And so we're going to just keep zooming in on more and more details about muscle contraction, but basically the myosin head naturally flexes. So if it touches the actin, it's going to flex and pull it. And then ATP is a very important thing. It's going to be used to detach the myosin. And where it grabs on again, it'll do the same thing and flex and pull the actin again. So this will keep happening as the muscle contracts until it's fully contracted. And when it's detached, it will let go and relax. So we'll see how that all happens. The fact that myosin heads are pulling the actin is called the sliding filament theory. It's how the filament is being pulled and sliding along. And the idea of the Z line is that those stripes that you see there in a sarcomere, one section of muscle, is a section that will actually shorten when the myosin heads are pulling on the actin. So you can see they're pulling until it's fully pulled, till this Z line comes so close to the end of the myosin we can't pull it any closer. So that sarcomere actually shortens as the myosin heads are pulling the actin closer. And so looking at Z lines, we can see under the microscope you can measure definitively how the muscle has contracted, because the sarcomere will shorten. The Z lines will come closer together. So zooming in on some of the structures, I'm going to go over to this diagram because it's the one you've got in your notes. But at rest, the actin here in green is wrapped around by, kind of reminds me of licorice, these ropes of tropomyosin, and that tropomyosin is covering all the binding spots. So this muscle is relaxed when the tropomyosin is covering the binding spots for myosin heads to bind to it, because as soon as it does, it's going to pull it. So the tropomyosin is covering the binding spots of the actin. And what will change is that these little heart-shaped troponin molecules are the spots where calcium can be attached to. And so if there's a message from your brain, your nervous system sends a message to your muscle to contract, because let's say you want to pick something up, the message will go to those muscles of the finger so that they will contract and coordinate themselves so that you can pick something up. What will happen is the calcium will get released out of the SR, the sarcoplasmic reticulum channels that are in the muscle cells. They'll release the calcium. The calcium then will bind to the troponin. And when it does that, the tropomyosin unclicks and moves away. It unclicks and moves away from those binding spots. So if it moves away from the binding spots, now you can see the myosin heads can bind to the binding spots, and what they're going to do is like crank and pull, because they naturally will flex and pull. So again, SR releases calcium, calcium binds to troponin. Tropomyosin, the licorice looking thing, pulls away. So as soon as the calcium clicks on, the tropomyosin pulls away, which allows the myosin heads to stick onto the binding spot of actin, and it's going to pull. So this myosin is going to flex and pull along the actin, and we will use ATP to detach those myosin heads. And if we still have calcium there, we will have that contraction. It'll pull again and pull it further, detach with ATP, pull it again. And this contraction can continue until the message is that we can relax our hand, in which case the calcium gets collected back up into the SR. And if calcium is gone, then this will cover up, tropomyosin will cover up the binding spots again, and our muscle will totally slide open and relax. So that is the process of muscle contraction. You can check out all of the video links to review that process of muscle contraction. Lots of little awesome animations to explain that. Just one more time, if we have a message that a muscle needs to be contracted, the sarcoplasmic reticulum releases the calciums, which will stick onto the troponin. When they stick onto the troponin, that will move aside this licorice-looking tropomyosin, in which case then the binding spots for this myosin is available to attach the myosin heads to the actin, and then they're going to flex and pull that muscle. And it takes ATP to detach it, and it can flex and attach and pull again. And that's how muscle contraction happens. If we remove the calcium, then this tropomyosin will stick back on, and we will have a relaxation happening at that point. And it's basically active transport that will grab all the calcium and put it back in the SR, and at that point the muscle will relax. Last topic for this section is about the energy for ATP being available. Has to come from somewhere, because when cellular respiration makes ATP, it says that ATP that's available at any one time is gone within a few seconds, like we talked about in our cellular respiration chapter. It doesn't last very long. We have to keep creating it. But if the muscles are working hard, they're going to look for or need other sources of ATP. So this is what they are. There's a creatine phosphate, which has ATP on it. And that is a source of ATP because we can, or a source of energy, because we can break off the 1-phosphate, attach it to ADP, and we can make an ATP that way. So creatine phosphate is one source. Obviously, aerobic cellular respiration, if we have oxygen, we're going to make 36 ATP for every glucose. So that is a source. If we don't have oxygen, then we can make at least 2 ATP through fermentation. So those are the 3 places ATP can be supplied to the muscle from. And it says that creatine phosphate is good for about 8 seconds of phosphate supply. And the most, of course, comes from aerobic cellular respiration, which we get most of our ATP for the muscles. It says here that a glycogen and fat can actually produce some ATP as well. So those can be kind of gone in through their own cellular respiration. Myoglobin, good to know. Remember, it's like hemoglobin. It carries oxygen for the respiration process. And it says that there's a lot of heat produced when muscles are contracting. That's why you feel so warm when you're working out. And that is also heat that's helping maintain your body temperature. Of course, fermentation is going to cause lactic acid buildup in the muscle. And what's going to happen is the lactic acid is making the sarcoplasm very acidic. And so that's why muscle cramps and muscle fatigue, muscle soreness result from too much fermentation. So what's going to happen after is that the lactic acid will have to get changed back into pyruvic acid in the liver. We talked about that before. And then it can be turned back into pyruvic acid and go through regular fermentation. So that's that. Okay. Last little bit here is that oxygen debt or oxygen deficit is going to cause that fermentation to be necessary to provide a little bit of ATP. However, it's not going to last very long because it's only a small amount, and it will create that lactic acid. They say in athletes, they build up more mitochondria in their muscle cells because of their constant use of their muscles. So they then possibly will have less lactic acid than someone who hasn't built up that same muscular strength. Replenishing oxygen debt, we'll need to get our creatine phosphate levels up, get rid of lactic acid, turn that lactic acid back into pyruvic acid in the liver. And so it says here that after a strenuous marathon, it may take a couple days on lots of carbs to get back the muscle stores that they had before. Alrighty, that wraps up 10.1, and I'll see you back for our last video lesson for the body systems.